Exosomes are these 500 to 100 nanometer spheres that are present in serum and other fluids. These are small little spheres that are basically released from all of our cells, and they have a message to communicate. My name is Vannberg. I'm a professor at Georgia Tech. The primary focus of my laboratory's research is genomics of infectious diseases. So we're focused on how the immune response basically responds to infections, and exosomes are part of that response process. What we find interesting about exosomes is this idea that they can transmit information to other parts of the body. Our recent paper shows that exosomes derived from stimulated cells, innate immune stimulated cells, are rapidly disseminated to the lymph nodes, and that has potential to actually help enable rapid delivery of therapeutics into the lymph nodes using exosomes derived from stimulated cells. And so these results really have some interesting implications for how we can actually stimulate the immune system to fight not only infections but potentially also cancer. There are many ways to purify exosomes, and they involve using antibodies to pull them down and other methods. We've tried many of these different methods, and the one that we really settled on is the classic approach of using ultracentrifugation, using Beckman centrifuges to purify them. The workflow is quite simple and actually does follow along with many other labs that do this procedure. We go through a multi-step process. And what happens is we're able to get very pure exosomes. We check exosome purity primarily looking at the markers CD81 and CD63, and these are classical markers for exosomes, these tetraspanins that are on the outsides of the exosomes. And we show using our ultracentrifugation approach, which is in line with many other research groups around the world, we show very high purity of exosomes there. Greater than 95% of the molecules there are exosomes. We use the Beckman Optimus centrifuge system because really it is known to be the best system out there. The ease of use and just the consistency of results is actually just excellent. What we really like about the Beckman Optima line is the fact that you have this record of exactly how the prep was done, and we can do it, the protocol, exactly the same way each time. And so that's why we've stuck to ultracentrifugation. One of the areas that I'm very excited to actually kind of monitor and hopefully be a part of is this idea that we could potentially use exosomes to target not only cancers but also potentially these pathogens that are evolving in the world. And so we've had some initial kind of thought into how we could use exosomes to actually load siRNA that would target things like Ebola virus, Zika virus, and other viruses that are important. And so the idea is to actually use exosomes now, start to create therapeutics even before these infections really take hold in human populations. And so that would be an astounding breakthrough, and it would kind of fit into the genomics of infectious disease work that we're doing in the lab.